Well, thank you everyone for joining us for this month's MIG seminar. It's a great pleasure to have Daniel MacArthur speaking to us today. He's now the Director of the Centre for Population Genomics at the Garvan Institute of Medical Research in Sydney and the Murdoch Children's Research Institute in Melbourne. Daniel, of course, has recently spent several years building up an extraordinarily successful large-scale genomics program and team at the Broad Institute at MIT and Harvard. Before that, he was in Cambridge in the UK, working at the Sanger after doing a PhD in Sydney. He joins us today from Sydney, not in stage 4 lockdown, thankfully, like Melbourne is, but he assures us it's grey and miserable there. So those of us stuck inside in Melbourne are not missing too much. Daniel will stick around afterwards to have a chat with early career researchers, so students, postdocs, anyone else who identifies as such. Please do hang around and take advantage of picking Daniel's brain, asking further questions and chatting about whatever you like. And the final thing I'll say is that Daniel's very happy to answer questions throughout the talk. So if you do have questions as we go along, please drop them into the chat. I'll keep an eye on that and I'll relay them to Daniel throughout his presentation. So with that, many thanks, Daniel, for joining us. We're excited to hear what you have to talk about today. Over to you.
Cool. Thanks very much, Davis. And I guess just to echo Davis's point, I would be very, very happy to take questions throughout the talk. So don't hesitate at all to either to interrupt or to post a question on the chat. It's always easier to clarify during the talk than have questions right at the end. So my condolences, obviously, to everyone who's stuck in lockdown down there. I can say from Sydney, it's obviously sad to see this, and also really challenging knowing this is likely on our horizon at some point in the not too distant future as well. So I hope you guys are staying safe and keeping yourselves morally and morally intact as you survive through the lockdown period. What I'm going to talk about today is I wanted to start with a little bit of background on the work that I had done in the past, amplifying a little bit on a couple of the points that Davis had made about my previous roles. Then introduce some of the work that I did in my previous position at the Broad Institute in Cambridge in Massachusetts. And then after that, I'll talk about some of the science that we did through the gnomAD project. And then after that, spend probably the last 5 or 6 slides talking about the new Centre for Population Genomics, some of the things we're planning to do in that centre, and obviously a shout out to anyone who's interested in working in this space. We'll have a number of jobs advertised in that centre, and I'll talk a little bit about how those will work. So I'll start with a bit of background on me. Let me just get my slides working first. So as David's mentioned, I did my undergrad and then my PhD in Sydney. I was based at the University of Sydney, and then did a PhD out at the Children's Hospital at Westmead. It was originally a full molecular biology PhD. I made a knockout mouse model of a gene called actinin-3 that my PhD supervisor, Catherine North, had already shown was potentially associated with athletic performance. There's a knockout. It turns out there's a loss of function variant in this gene that knocks out its function in about more than 1 billion people worldwide. They're homozygous knockouts for this gene. And we were able to show both in the knockout mouse and also through association studies in humans that the loss of function of that gene has a really profound effect on muscle function and means that fast muscle fibers become more like slow muscle fibers in the people who are missing that gene. That project got me really interested in the question of other loss of function variants that could be found in the human genome, so other naturally occurring mutations that knock out the function of human genes. And so I was very keen to go on and do a postdoc where I could explore this question further, and ended up settling in the lab of a guy called Chris Tyler-Smith at the Wellcome Trust Sanger Institute in Cambridge. And there I was able to explore these loss of function variants in really the first large-scale data set of sequenced human genomes. It's actually kind of laughable thinking about this as a large-scale data set now. At the time it seemed huge, but it was 185 human genomes that we'd sequenced as part of the pilot phase of the 1,000 Genomes project. So we looked at those loss of function variants in those genomes, and the key finding from that project was that all of us carry probably about 100 to 150 true gene disrupting variants that actually knock out the function of one or more of our genes. And most of those are in any one individual are actually relatively common, and they affect genes that are dispensable. But of course, there's always the possibility that those loss of function variants will land in a gene that's absolutely critical for survival, and that is the basis of many different classes of severe Mendelian diseases. So after wrapping up that postdoc project, I moved on to start a lab which was a joint venture between Massachusetts General Hospital and the Broad Institute across the river in Cambridge. And in my group, I really focused on two distinct areas. The first was using genomic approaches to improve the diagnosis of rare disease patients. And then the second was setting up reference databases of genetic variation. And the reason that we needed to build those reference databases is because it became clear as we started to do approaches like exome sequencing and genome sequencing on the rare disease patients, mostly collected through collaborations with groups around the world, including a number here in Australia. It became very clear that firstly, in order to make sense of the variants we were discovering in those rare disease patients, we needed to be able to look at those in the context of databases of general population variation, in order so that we could say whether or not that variant was common and therefore whether it was likely to be associated with rare disease. And then the second thing that became clear was that the existing databases of variation, at the time these were things like the 1000 Genomes Project and the NHWI's exome sequencing project, were both too small and relied on a pretty antique variant calling methods, and therefore didn't really serve the needs of the clinical genomics community to interpret variation. And so we set about really pursuing these two research angles in parallel. On the rare disease side, working with Heidi Ream, we established an NIH-funded Centre for Mendelian Genomics. And over the subsequent 5 years that centre ran, we were able to apply exome, genome, and in some cases RNA sequencing to more than 6,000 families affected by a whole variety of different rare diseases, ranging from severe muscle disorders through retinal diseases and non-syndromic intellectual disability. We diagnosed about a third of the patients in that cohort, and we were able to return those diagnoses back to families through our collaborators. And we identified over 300 candidate novel disease genes, many of which have now been published, and there's a number still in the pipeline for functional and clinical validation. I'm not really going to talk more about that work today, but just wanted to mention it sort of as a background to the work we did on the reference database side of things. And then the second half of my lab focused on that challenge of building these big databases of normal variation. And here we were able to take advantage of the pretty unique situation that existed at the Broad Institute, where because it was such a large sequencing facility, much of the world's sequencing of consortia focused on common complex disorders had actually been done through the Broad, and there was a significant willingness to think about sharing genetic data. So the opportunity for building these databases was clearly huge. We knew at the time that many tens of thousands of exomes had been sequenced, and this was an effort that we started back in 2012. These days, it's difficult to get a real sense of how many humans have actually been sequenced, but it's certainly the case that at least 3 million, likely very significantly more than that, people have had either their exomes, which is the protein coding bits of their genomes, or their whole genome sequenced around the world. And those sequencing efforts have been done in the context of a whole variety of different projects, many of them in academic settings, but increasingly we're seeing more and more data generated in the context of industry-driven collaborations like the UK Biobank project, for instance, where the sequencing is being funded by a pharmaceutical company consortium, or other cases where companies like Regeneron or Amgen, for instance, are building up their own proprietary massive databases of genetic variation. So these data in aggregate are an amazing resource for understanding the patterns of variation across the human genome. And in an ideal world, we figure out a way of bringing all of them together and analyze those in the context of clinical information about people as well. But of course, in real life, aggregating that data is not straightforward. There's lots of different problems that we would need to tackle to make that feasible. Some of the problems are pretty mundane. There's things like the fact that it's hard to move and process data. And this is particularly true when the data is scattered all across the world, although that is becoming a lot easier in the world of more pervasive cloud computing, where it becomes easier to store the data in a single place and move our analysis processes to that data. There's a second problem that's actually much more significant, which is the fact that the samples underlying these sequencing projects have typically been collected for decades, and often they've been collected at a time where consent was done before there was really any thought put into the idea of large-scale genomic data aggregation. So the consenting and the data use permissions often end up being the major obstacle to aggregating and sharing data. And I can tell you that in the course of the projects that I'll talk about later, a terrifyingly large fraction of our time was actually spent dealing with ethics and data use permissions problems. There's, I guess, a set of related problems which are around the fact that the people who generate that data don't always want to share the data. Sometimes that's because there's commercial restrictions associated with the way that data was generated. Sometimes even in the academic setting, we have people who are not interested in making the data available. So that's always a problem. And then there's a final technical challenge, which is the fact that every group that generates human sequencing data tends to process and analyze it using a slightly different pipeline for interpreting variation. And that means that if you were to just take the outputs of all of those different pipelines and try to jam them all together, you would end up with a data set of variation that was dominated by boring technical differences between pipelines, as opposed to interesting biological differences. So if you want to be able to get stuck into the biological challenges, you need to have a collection of human variation data that has gone through exactly the same pipeline and where you've made a real effort to harmonize the underlying data. And that ended up being another enormous fraction of the work that we did in aggregation. So the work that we led at the Broad Institute in building these big reference databases proceeded in 3 phases. Beginning in 2012 and then culminating in 2014 with the first public release of the data, we had an effort called the Exome Aggregation Consortium, or EXAC. As the name implies, this was focused entirely on exome sequencing data, and there we were able to collect just over exome sequencing data from just over 60,000 people, pull that together, do joint calling across the entire data set, and then make that data publicly available at American Society of Human Genetics meeting in 2014. And then pretty much at exactly the same time, we started work on the next call set, where we also wanted to include whole genome sequence data. We changed the name to reflect the fact that we've added genomes to this data set. It's now called the Genome Aggregation Database, or gnomAD. And the first release of that, which came out in 2016, basically doubled the number of exomes that we had. And we were also then able to include nearly 16,000 whole genomes. And then in October last year, we were able to release an updated version of the whole genome section of that database. This is called gnomAD v3, and that contains information from over 70,000 whole human genomes. And that's really very useful in terms of being able to look at the distribution of variation outside the protein coding regions of the genome. So just a few points about these collections. The first is that to a large degree, these projects became as much about data science and engineering as they were about science, although we were then able to go on and do some really cool science with the resulting data sets. And that's because the underlying data are huge. So in aggregate across these 3 collections, we had to process something like 8 petabytes of raw sequence data that flowed into each of these projects. The way that we were able to get these data was not by generating it ourselves. The gnomAD, EXAC and gnomAD projects are data parasites. We don't generate our own data. We take advantage of data that's been generated by other groups. And that mostly comes from case control studies of adult onset complex disorders. So these are things like type 2 diabetes, heart disease, neuropsychiatric conditions like schizophrenia or bipolar. And in those cases, we incorporate both the cases and the controls for those individuals together into these data sets. What we do take as much care as we can to avoid is including individuals who are known to have severe pediatric diseases. So for instance, people who have autism or a whole variety of different pediatric genetic conditions or pediatric cancer, we try to exclude those from the database. And the reason for that is that we want this to be a reference database that can be used to compare severe genetic disease patients against. So we don't want it to be enriched for the alleles that are associated with those diseases. We have taken, for each of these 3 releases, what we did is we took all of the underlying raw data at the read level. We processed that data through the same pipeline, and then we did what's called joint variant calling, which means that for every single one of these individuals, we perform variant calling across all samples simultaneously to ensure that we have, at each of those sites across the genome or the exome, that variant has been called the same way in everyone. And that helps us be confident that the resulting allele frequencies are well calibrated. That is, we've called the same site the same way across the entire sample. And then a final point to make is that we focused as much as we could, although of course here we're constrained by the fact that we're dependent on other people's data, but as much as we could, we focused on building in diverse ancestries into these databases. And although we're limited by the fact that most of the genomic data that's being generated around the world at the moment is of European ancestry, and that continues to be a huge problem that I'll come back to later, in the gnomAD dataset, we were able to include more than 10,000 samples each from South Asian, Latino, African and African American, and East Asian ancestry as well. So the gnomAD dataset at the end is definitely by no means a comprehensive sampling of human genetic diversity, but it does give us a pretty good triangulation for a number of the large populations, at least that exist in the US setting. And it gave us a reasonable reference data set that we could use to compare for many of our rare disease patients. So at least it's a starting place. For each of the releases that we've done with EXAC and gnomAD, we've tried to make that data available as quickly and openly as possible. So we've done that in a number of different ways, but one of the approaches has been through the development of these browsers. The most recent one is the gnomAD browser. I put the URL up in the top left-hand corner. And that's been the work of a number of engineers over the years, but more recently, Nick Watts has really taken over the running of this browser and pulling together a whole bunch of different features. And that effort has been kicked off and then overseen by Matt Solomonson as well. So the browsers provide all sorts of information about the data that we find in gnomAD. So for instance, you can see the coverage of individual components of a particular gene. There's information about the transcript expression of that gene. You can look up known pathogenic variants. And then in this table down the bottom here, we have a long list of every genetic variant that we've discovered in that gene in the gnomAD dataset. And you can click on those variants to learn more about how common they are in different populations, all the functional annotations that we can find associated with those variants, and also how confident we are that the variant is actually real. And for most of the variants in gnomAD v2, we also make available the raw read data supporting that individual variant. So you can actually take a look at the read data itself and see if you're convinced that the variant is actually real. So because we release these data widely and because of the scale of the databases, it's been amazing to see how quickly and widely used they've been. This here is the page views from the EXAC and gnomAD browsers over the years since we launched the first EXAC browser back in 2014. So that's the EXAC browser in blue, and then the gnomAD browser as it slowly took over in green. And this is the weekly page view count. You can see a bunch of stuff from this plot. Firstly, you can see kind of reassuringly the fact that every year around about the Christmas break, genomicists kind of put down tools and stopped using the gnomAD browser for about a week, which is always good to see. And we've been actually tracking the usage of the data through 2020 with some interest, and it seems to have basically plateaued. So we're not seeing growth this year, but we are seeing that despite the global issues imposed by COVID, there's still pretty heavy and ongoing use of the database. And that's mostly being driven by ongoing use by clinical genetics groups around the world, where obviously their diagnostic work is continuing regardless of the pandemic situation. So we don't know exactly how many families the gnomAD and EXAC databases have contributed to the diagnosis of, but by talking to US clinical labs, we know that at least 100,000 patients have received diagnoses where the EXAC and gnomAD databases have contributed to the interpretation of those variants. So they really have become a widely used resource in that setting.
Daniel, a couple of questions
go ahead, David.
Come through. So Joe McSee writes, are the processing steps available, and do you have to use the same processing to compare a sample to the database?
Yeah, great question. So we have released, so all of the code we use in gnomAD is available. We use open source tools. So the underlying code that we use for processing is basically following GATK best practices established by the Broad. So long as you keep using those ongoing best practices, the data that you generate should be broadly compatible with gnomAD. Although it's always worth checking in with the Broad team to just make sure that the version of the pipeline you're using is closely enough aligned. all of our downstream processing involves combinations of the GATK and also a software processing tool called Hail, which is a platform for large-scale analysis, and we actually now use Hail for our joint calling process as well. All of that code you can get through the gnomAD GitHub repository, so that's available through there as well. In terms of exactly how close the processing has to be, I think so long as you follow GATK best practices and in particular filter your variants carefully, you will have a data set that is at least broadly compatible with gnomAD. Now that said, we think that the value of gnomAD is primarily as a reference data set. So that means if you're analyzing a rare disease patient, you can look up a particular variant and see its frequency in gnomAD. We urge great caution in using it as a control data set for statistical genetic analysis. So for instance, if you've got a set of exomes that you've sequenced for a complex disease, we don't think that gnomAD is a replacement for sequencing a set of controls for gene discovery in that particular setting. There, you're better off basically making sure that you have fully technically matched controls that are incorporated as part of that project. And gnomAD is best suited as a reference data source rather than a substitute for those controls. And there's lots of reasons for that, but one big thing is obviously because we only release the aggregated version of the data, it means you can't correct for things like population stratification or batch effects in analyzing those data.
Thanks. And just one more for now. Gokchen Eraslan asks, were any whole exome sequencing samples banked so that resequencing them using whole genome sequencing is possible, or are all samples destroyed after sequencing?
This is a total mix of samples. And again, the data that we pulled into gnomAD really comes from a whole variety of different consortia and sources. But most of the data that was, because most of the sequencing was done at the Broad Institute, the Broad maintains a biobank, which I think is now up over 1.5 million samples of all of the DNA samples that they use there. And that means that for many projects, those consortia have actually gone back over time and redone sequencing with whole genomes as well. But it's a patchwork across the data set.
Okay, final question just now, and then I'll let you get back to your slides. Dr. Shalou, whose name is then obscured, the pediatric database, disease database depleted in gnomAD, is that banked separately or publicly available?
Oh, yeah. Sorry, I wasn't very clear on how to describe that. So what we've done there is we, in defining the samples that flow into gnomAD, we try to exclude as much as possible the individuals with pediatric disease, but those samples are then just not included. So it's not that we keep a separate database that is composed of pediatric disease. For thinking about databases of rare disease variants, we basically would encourage people to use resources like the ClinVar dataset. And we try to make those types of things available through gnomAD, but that's really a different set of questions. The questions about which variants are present in patients we see as quite distinct from what gnomAD provides, which is a set of variants from individuals who are not rare disease patients. So we're trying to really provide that sort of population reference side of the equation here.
Yep.
Cool.
Thank you. Great.
All right. Thanks, Davis. That's great. All right. So for those of you who are interested, so with this big collection of data that's come out from gnomAD v2, there's obviously a ton of different scientific analyses that you can do. And much of the last couple of years, 3 years really, we've been focused on quality control of that data set and then analyzing that this big collection of exomes and genomes from a whole variety of different angles. And that culminated in May this year with the release of a set of 7 papers describing those analyses. If anyone's interested in reading more, I've just put little short links up here to each of the individual papers. I'll only touch on results from a couple of these, but there's a lot of stuff to cover here. There's also a nice little collection that Nature put together, which has a news and views by Gianna Church that describes some of the ways that the gnomAD dataset has been used, which I really liked. So feel free to take a look at all those things. I'll just mention briefly that these papers describe, firstly, the generation of data, both for SNPs and indels in this first paper here, and then for structural variation in the second paper. So that's large deletions, insertions, or rearrangements of DNA. And that work was led by Michael Talkowski's group. We also talk about the analysis of loss of function variants. And I mentioned right at the start of my talk, this has been a long-standing interest of mine, is these variants that are predicted to cause the disruption of genes, and I'll come back to those later. We spend a fair bit of time talking about the way we can use genetic variation to identify and potentially validate human therapeutic drug targets. And that's in a sort of overall review by Eric Minikel, which is published here. And then secondly, a case study where we looked at loss of function variants in a gene called LRRK2, which is a known therapeutic target for Parkinson's disease. And in that paper, which we published in Genome Medicine, there was a description of basically what we're able to show there is that individuals who are naturally deficient in LRRK2 appear to be healthy, suggesting that partial reduction of LRRK2 function, which is the goal of therapeutics for Parkinson's disease in people with gain of function mutations in LRRK2, that's likely to be a safe or at least well-tolerated therapeutic strategy. And then we had a number of papers that were focused on the ways you can use this data set to improve the interpretation of genetic variation in a variety of clinical contexts. So happy to answer questions about some of those areas later. But today I'll mostly just focus on a few of these areas, the analysis of loss of function variants, and then some of the ways we can use these to understand potential drug targets. So firstly, I just wanted to mention this target of mine, loss of function variants, or actually what we call predicted loss of function, or PLOF variants. The gnomAD dataset contains a huge amount of variation, nearly 250 million variants in total. And of those, more than 500,000 are predicted to be loss of function variants. So that is, if you naively just said, based on where that variant falls within a gene, what effect it's likely to have, those variants are either predicted to result in the gain of a stop codon, to disrupt an essential splice site, that is the two bases at either end of a spliced exon, to create a frame shifting insertion or a deletion in the middle of a coding sequence, or cause a large deletion that's predicted to remove more than 50% of the coding sequence of that gene. Now, the rate of discovery of those loss of function variants continues to increase. And so this is a plot we generated, this is from Konrad Kaczewski, where he downsampled the gnomAD exomes. So that's starting on the right-hand side of the plot. We then downsample moving left. So you can see basically how many new loss of function variants we gain as we add each new exome to the data set. And what happens there is that we get an initial early gain where we find all the relatively common loss of function variants. Most of these are in pretty uninteresting genes. And then after that, we enter a roughly linear phase where we now discover about one new loss of function variant for every exome that we sequence. And that trend is predicted to continue to be roughly linear right up into the tens of millions of exomes, basically until we start hitting the point where that starts to tail off, because all of the remaining loss of function variants are so deleterious that they tend not to be represented in the human population. So that suggests that we can the more we sequence people, the more of these loss of function variants we'll discover, and the more we can use them to interpret the function of human genes. But of course, the impact of those loss of function variants will vary enormously depending on which gene they happen to fall in. And in a magical world where we could array genes along an axis based on their loss of function tolerance, you might have a spectrum that looked a little bit like this, where on the left-hand side, you have genes where any loss of function variant is catastrophically damaging. And in the most extreme case, you might have a situation where losing even one copy of that gene results in embryonic lethality. So you'll never see a living person who has a true loss of function variant in that gene. So those are on the left-hand side. In the middle, we have the genes where disruptive variants result in clinical conditions, a whole range of different severe Mendelian diseases, where some of those will be haploinsufficient, that is, knocking out one copy is enough to cause disease, and some of those will be recessive, meaning that you actually need to knock out both of those copies before you get a severe disease. And then on the right-hand side, there are genes that are more tolerant of inactivation, but where there could still be some effect. So in some cases, knocking out one copy might be enough to just increase your risk of type 2 diabetes by 10 or 20%. And then on the far right-hand side, there is likely a tiny fraction of the human genome where knocking out that gene is actually beneficial. That is, it has some effect that results in an increased lifespan, or at least protection against a subset of human diseases. And we know of a handful of these genes, like PCSK9, for instance, that protects against high LDL cholesterol and it significantly reduces your risk of heart disease if you have one copy knocked out. So the genes on the left-hand side of the spectrum, although they're the ones where there's the most extreme effects when you knock those out, these are actually the genes that we know the least about in humans. And that's because the individuals who have loss of function variants in these genes are typically not seen. They disappear during embryogenesis and are not actually born. But we can actually infer the presence of those genes by studying the signature that's left behind in EXAC and gnomAD by the absence of the variants that we would otherwise see if those people did actually exist in the population. So to do that, we, working with Mark Daly, and this has been work that was initially led by Caitlin Samoka, a PhD student in Mark's lab, and then by Konrad Kaczewski in my lab. What we did in this set of projects was to develop a statistical model that allowed us to predict for any given gene in the genome how many loss of function variants we would expect to see under a random mutational model. So that is, if natural selection was not acting and all you had was mutation generating loss of function variants in that gene, how many loss of function variants would we expect to see in a sample size of n, where n was the size of whatever reference database we happen to be looking at. And using that expected model, we can then compare that with the number of loss of function variants observed in that gene in EXAC or gnomAD, and look at the ratio of the observed to the expected number to get some handle on what fraction of loss of function variants are actually missing from the population in that gene. And that's a measure of the degree to which natural selection is acting to remove those variants from the population. And to give a statistical estimate of that, Konrad developed a metric called LERF, which is the loss of function observed to expected upper bound fraction. It's basically the upper end of the 90% confidence interval of that ratio. And that corrects for the effects of small genes, which might have very, very low observed to expected ratios just because the number of expected variants is tiny. And that gives us this spectrum that you can see on the left-hand side over here, where genes that are highly depleted for loss of function variants are shown here on the left, and genes that have lots and lots of loss of function variants, potentially even more than what we would expect to see, are found over on the right-hand side of the spectrum. And that corresponds roughly to the cartoon spectrum that I showed on the previous slide. So here we have a measure that we can use to actually put genes along that axis. Although one important caveat that I'll mention here is that because natural selection acts primarily in the heterozygous state, what we're mostly seeing here is how sensitive a particular gene is to heterozygous inactivation. That is, to having a single copy knocked out in an individual carrier. So what happens when we put normal? So we can use that spectrum to look at a whole bunch of standard gene lists and see where they fall, and they mostly seem to fall in pretty sensible places. So, for instance, genes where we know that knocking out a single copy causes a severe pediatric onset disease, almost all of those genes are found towards the left-hand end of the spectrum, and the scattering of genes that are found over here on the right are all very small genes, where the LERF metric basically pushes those over to the right because the confidence intervals are so large. So that's reassuring. Recessive disease genes fall into the middle of the spectrum, and that appears to be driven by selection against carriers of those recessive disease mutations, which is pretty interesting. So that suggests that although the standard clinical manifestations of these recessive diseases are only seen in homozygotes, there is at least some measure of selection for many of these genes against the heterozygote carriers as well, although that often won't be clinically visible. And then for genes that are largely dispensable, and here's one family of those genes, olfactory receptors, where knocking out one of those copies will just mean that a human is not as good at smelling a particular compound, but typically won't be life-threatening. Those genes tend to be found on the right-hand end of the spectrum. So they are basically acting neutrally with respect to the number of loss-of-function variants that they're accumulating. And these metrics also correlate nicely with a number of other biological measures of gene function. So for instance, genes that are highly constrained have significantly more protein-protein interactions than genes that are not constrained. They also tend to be expressed in more tissues compared to the rest of the genome. And most importantly, this LERF metric allows us to prioritize genes that are involved in severe disease. And we've now applied this approach to a lot of different diseases, but I'll just show one example here. This is work that was led by Jack Kosmiski in Mark Daly's group. What Jack did was to pull together from his lab's own data, as well as publicly released data sets, a collection of de novo variants identified in intellectual disability or developmental delay cases, and then compare that to a set of trios where the child was unaffected, and look at the rate of these de novo variants in the patients versus the controls. And then compare that rate across different gene sets relative to their level of loss of function constraint. And what he showed is that for synonymous variants, de novo synonymous variants shown here in gray, this distribution is completely uniform. That is, it doesn't matter how constrained a gene is. There's no difference in the rates of de novo synonymous variants between cases and controls. But for de novo loss of function variants, that is very non-uniform. Almost all of the genes where de novo loss of function variants are seen in cases, almost all of the signal of enrichment is found in the most extreme 10% bin of the loss of function constraint spectrum. And that tells us that even by looking at gnomAD, where we're looking at a set of individuals who are not affected by severe disease, we can identify a set of genes that are vastly more likely to be involved in a whole variety of different conditions. And this same signal of enrichment in these most highly constrained genes shows up in a variety of other conditions, including autism and schizophrenia, a variety of dominant Mendelian diseases, and interestingly also appears to show enrichment in common complex disorders as well. So there it looks as though these more common regulatory mutations nearby these genes are actually more likely to cause a whole range of common conditions and traits in the UK Biobank data as well. So that's what we can learn about this left-hand end of the spectrum. I just wanted to spend a little bit of time talking about the right-hand end of the spectrum and understanding the impact of loss of function variants on those genes, because that can be very important. Although these genes are not necessarily involved in severe human disease, understanding loss of function impact can help us to understand what would happen if we were to therapeutically target those particular genes in a whole range of different conditions. So here what we've been thinking about is the idea of a human knockout project, which would basically be a catalogue of individuals who have a complete inactivation of each of the genes in the human genome, and where we could then link that knockout genotype with individual clinical phenotypes. So there's a number of things we need to be able to do in order to build such a project. The first is we would need to make sure that the loss of function variants we were discovering were actually real gene disrupting variants. And that turns out to actually be quite complicated for a variety of reasons. These loss of function variants are actually enriched for all sorts of different artifacts, and we have to spend a lot of time filtering and curating those variants to get a good high confidence set of loss of function variants. We built a lot of tools for doing that. Again, I'd be happy to talk about those in more detail later, but won't go through them today. Those are our software tools, though, are publicly available if anyone's interested in using those. And just to give you some examples about how important this approach is just two genes. The first is the HTT gene, which encodes Huntington, a gene where severe gain of function mutations result in dominant disease. In this gene, it turns out there's a lot of allegedly loss of function variants. In fact, in total, 6.2% of all of the copies of HTT in gnomAD carry at least one loss of function variant, which seems really implausible for such an important gene. And actually, this y-axis is on the log scale. Most of those variants are actually found in the first exon of the Huntington gene, which is pretty suspicious. And it turns out that, in fact, all of those alleged loss of function mutations in the first exon are found in the polyglutamine tract. That's the same polyglutamine tract where expansion actually causes Huntington's disease, and they're mapping errors, so they can be easily removed. And there's a variety of other types of artifacts that we can filter out using our methods as well. But some true loss of function variants remain, all of them rare, and the combined loss of function allele frequency in this gene after curation goes down to about 0.01%. So it's a huge number of artifacts get removed by our filtering. Here's another even more extreme example. This is MAPT, which encodes the tau protein, another critical gene involved in neurodegenerative disease. The combined loss of function allele frequency in this gene is 14%, and that seems completely implausible. And in fact, it's entirely wrong. It turns out that most of the candidate loss of function variants in this gene fall in a set of genes shown, a set of exons rather, shown here in white, which are not expressed in the brain. And all of the other loss of function variants we identify in this gene turn out to be either annotation artifacts of various different kinds. So after applying our annotation approaches, as far as we can tell, there's not a single individual in gnomAD who carries a true loss of function variant in this gene. So it's completely depleted for true LOF variants. And that really emphasizes the importance of thinking about exon level expression in interpreting the variants we find in genes. And one of our gnomAD papers by Beryl Cummings is focused on that process. So the second thing we need to do is ensure that we can identify individuals who are more likely to be homozygous for loss of function variants. And really there's two strategies that we could follow to identify such individuals. The first is we could look at populations that have gone through what we would call population bottlenecks, where a relatively small number of founders with a restricted genetic set of haplotypes have gone through a type bottleneck and then expanded up to a relatively large modern population. The benefit of those populations is that in a relatively small number of genes, there will be loss of function variants that happen to be present in that founder population that have surged up to high frequency in the modern population, and therefore there'll be a significant number of human knockouts for that gene. And we see that, for instance, in the distribution of diseases across these bottleneck populations. The downside is that each population will provide those loss of function variants for only a small number of genes in the genome. So that means you'd need to study many, many populations before you could start to have a reasonable probability of finding a loss of function variant in a particular gene that you cared about. A second strategy you could follow is to work with populations with a high fraction of consanguineous individuals. So that is individuals whose parents are related to one another due to a number of different cultural practices. And in those groups, the number of homozygous loss of function variants turns out to be extremely high, about 30 times higher than in a randomly ascertained European population. And those are scattered across the genome. So if you're interested in identifying a single knockout individual for a particular gene, these consanguineous populations are extremely well powered, as I'll show on the next slide. The one challenge, though, is that for each of these genes, sorry, you may only be able to find knockouts in one or a handful of individuals within a small family. So statistical power can be a challenge there. Just to show you how much better power we have for identifying these human knockouts in consanguineous populations. This is modeling work that's been led by Eric Minnickel, was published in his paper, short link down the bottom there. What he did is to take the roughly 10,000 genes in the genome where there is at least one loss of function variant in gnomAD, but we don't yet know what the loss of function phenotype is for that particular gene. And he used that to model the way in which we would expect the growth of discovery of knockout individuals as we sequence more and more samples. The x-axis on this plot is very strange. It's a log axis that goes from 1,000 individuals on the left-hand side to the entire world's population on the right-hand side. So basically you can see what our discovery curve would look like as we started to approach sequencing the entire population of the world in black, or the entire consanguineous population of the world shown here in this brown color. And the discovery curves are remarkably different if we were to look in a randomly ascertained European population, shown here in black. We would need to sequence something like 30 million individuals before we were able to identify a single knockout in about 50% of those genes. 30 million is not completely insane, but it's going to take us a long time before we get there. In contrast, we would need to sequence about 100,000 individuals of consanguineous parentage to get to the same number of genes knocked out. And so that's been the driver for a number of projects around the world focused on recruiting and doing exome sequencing and genotype-based recruitment of consanguineous individuals. I've been involved in a number of these. I think the most successful right now is the East London Genes and Health Project based in the UK, where more than 50,000 individuals have already been recruited. And we're currently working on building up the resources for exome sequencing of up to 100,000 individuals funded by the Wellcome Trust. All of those individuals are recontactable, and there's also medical records. So it's linked, the underlying data is linked to longitudinal electronic health records in the NHS, and that work is ongoing. It's been led by David Van Heel, in particular, working with Richard Trembath, Matt Hurls, and Australia's own Hilary Martin, now based at the Sanger Institute. So that's a fantastic project, and I think it would be, it really provides a phenomenal model for building out these human knockout projects around the world.
Daniel, can we sneak in a couple of questions? So Lauren Akerson asks, regarding the lofty filtering that you show those examples of, do those artifacts or artifactual variants still appear in gnomAD, the database, or are they removed once they're identified?
Yeah, great question. So we flag them in gnomAD. If you look at the list of variants underneath a particular gene, you'll be able to see a little red flag next to some of those loss of function variants that indicates that they've been lofty filtered. Now, lofty will catch a subset of the easily automated types of annotation artifacts that we can find. We have found, particularly for high value genes, that we also need to do manual curation. We've actually done that now for about 4,000 candidate loss of function variants found in gnomAD across a whole range of different genes. And that manually curated data is being dumped into gnomAD as we generate that as well. But that's obviously it's unevenly scattered across the genome. Only a certain handful of genes have been fully annotated in that way.
Excellent. Johnny Choi asks, can the data tell us anything about CNV cases where there may be additional copies of a gene?
Oh, right. So duplication situations. So I think these are really interesting. The closest we have to a global model for gain-of-function mutations. The challenge, of course, is that duplications are hard to call. We do have a systematic call set of duplications that's available on our gnomAD v2 dataset. That's our 15,500 human genomes that was generated by Michael Tarkowski's group. But it's still underpowered for most analyses. And the next phase of analysis that Michael is again leading is to do systematic calling of copy number variants across our 70,000 individuals from gnomAD v3, all from whole genome sequence data. And I think it's only with that scale that we'll be able to start to say something sensible about the gain-of-function tolerance of individual human genes. One interesting finding, though, from that early analysis is that it looks like genes that are intolerant to loss-of-function variation also appear to be systematically intolerant to gain-of-function variation as well. So they're depleted for duplications too. So that suggests that in general, they're just very dosage-sensitive genes.
Excellent, thanks. More questions are coming in, but I suggest maybe we let you.
All right, I'll keep
going. And there'll be more.
Perfect. Okay, sounds good. So just a quick sense of what's coming in the gnomAD project. So in early 2021, we have 2 call sets we're hoping to release. The first will be a new exome call set on build 38 that will have over 500,000 exomes. And for the first time, that will include data from the UK Biobank exome project. And that's incredibly exciting because it means we can actually link the data we're finding in gnomAD with the deep clinical data that's been collected on that resource. And then the second release I've already mentioned, and that's led by Mike Tarkowski's group, where he'll be looking at structural variants and also repeat expansions on over 70,000 whole human genomes. And that will be really powerful for digging into the more complex structural variation across the genome. There's lots of things, scientific questions we're interested in answering. I won't go through those in detail, but these often revolve around constraint, expanding the idea of constraint outside the coding regions so that we can explore it in non-coding regions as well, using our whole genome sequence data. But then also with this huge new bolus of exome sequencing data, we will have vastly more resolution for understanding loss of function constraint, but also for identifying individual regions of genes that are depleted from missense variation. And we've already seen that that be useful in identifying particular pieces of the human genome that are most likely to be involved in severe disease. So there's a lot more work left ahead on that. Now, as I'll say in a minute, I left the Broad Institute at the end of 2019 to take up this new position in Australia. The data set around gnomAD is baked into the Broad Institute. So in departing, I needed to make sure I could hand over that project into safe hands. And fortunately, there were many safe hands available. So the gnomAD is now co-directed by Mark Daly, who brings in a wealth of experience on the statistical genetics front, and then Heidi Ream, who is, of course, very focused on clinical genetics and genomics. Both of them share the same philosophy of widespread data sharing. We have a scientific steering committee that includes myself, as well as many other individuals involved in analysing the gnomAD dataset from many different angles. So I think we're in a good position to keep the project going on, and I expect many future releases and lots more science to come from that project. So I'm staying engaged in that, but have now physically moved over back home to Australia to build this new Centre for Population Genomics that Davis mentioned. I want to just say a few things about why I did that. The first reason I moved back to Australia was entirely personal. I miss this place intensely, and although I grew up in Sydney, I have over many years and many visits come to deeply appreciate all of the pleasures of Melbourne as well. And so in returning to Australia, I was looking forward to being able to do something that really spanned these 2 fantastic cities. I'm still looking forward to doing that, although it's obviously going to be a little while before I can travel back and forth between the 2 of them. And there was also a lot of scientific drivers for coming back here. From a population genomics perspective, Australia has a remarkably diverse population, and including a significant number of communities that are totally unrepresented in reference databases like gnomAD, which I'll come back to later. But about which we know almost nothing from a genomic and genetic association perspective. So that's a real advantage. We have a healthcare system which, of course, has its flaws, but is, as we've seen tragically over the last few months, is vastly better equipped to organise large-scale efforts than the equivalent system, for instance, in the US. And so that's a real advantage both for research and also for translation. We have an interesting population size here. It's big enough that there's actually power to think about association studies for many diseases, and also to study a significant number of rare diseases. But Australia is also small enough to be nimble and think about full national-facing efforts across a wide range of different diseases. And then 2 things that have changed a lot since I left 11 years ago to work overseas. Firstly, there's now a significant funding commitment in the genomics space, although it's never enough, of course. And secondly, thanks to the efforts of the Australian Genomics and other collaborative efforts, there's now a really strong sense of a nationwide collaboration particularly in clinical genomics, but in genomics in general, and data sharing across the country. And that has been a tremendous change in the time since I left. So working with a number of groups, we were able to put together a proposal for a new Centre for Population Genomics. This is now jointly based at the Murdoch Children's Research Institute in Melbourne and the Garvan Institute in Sydney. Our goal is to build a set of tools and resources that will enable the equitable deployment of population scale genomic medicine in Australia. Our goal here is to build a single unified team, so to not have a Sydney group and a Melbourne group, but a single group that works across both sites. And in some ways, the COVID-19 situation has made that easier because we're launching in a fully remote mode where all of us are joining our meetings entirely virtually. And I think that's helping to build a single culture across those two sites. And we'll have many shared scientific projects across the two sites as well. This is the structure of the centre. We've been in the process of hiring a number of senior individuals across over the last couple of months. So we're led by an executive committee, involves me, Catherine North, and Chris Goodnow at the Garvan. We have 4 core teams within the centre: a project management team, an inclusive genomics team that heads our community engagement efforts, a software team now led by Leo Grunschloss, who's joining us from Google Sydney. We're building out an analysis team that we focused on managing and building out new analysis methods for data. And then we'll also be appointing associated faculty members over the next couple of years as well, both in Sydney and in Melbourne. We filled a number of these senior roles with people both in Sydney and in Melbourne. But I did just want to mention we're actively recruiting at the moment for 2 positions, the head of our project management team, which will lead the flow of samples and data across the entire centre, and also the head of our analysis team. So this would be an opportunity to recruit and lead a team of computational biologists and statistical and population geneticists working obviously on really large genomic data sets. So if you or anyone else are interested in applying for those roles, feel free to drop me a line. Some of the principles underlying the centre, we want to do this obviously at a very large scale. The goal is to be highly collaborative, as with all of the work that I had done previously in Boston. We'll be open in everything we do. So all of our software will be open source, our data widely available. All of our papers will be pre-printed and published open access. And from the outset, we are pushing the importance of diversity, both in the science that we do, and I'll talk about that in a second, but also, of course, in building out our team. So the scientific goals of the centre are firstly to create a data platform that allows us to store and analyse data at scale. We're still working out the details of how that will work. That will be almost certainly a cloud-based platform to allow for scale. It needs to be able to handle thousands to tens of thousands of genomic data sets. It also needs to fundamentally be open source and redeployable and reusable by other groups around Australia and elsewhere. And so we'll be partnering with other groups to modify existing open source code to make that platform feasible. We're still engaging with a number of stakeholders in Australia to figure out exactly how that will be built. And then from that data platform will flow our scientific projects, building a new reference database of human variation that I'll talk about a little bit more, applying genomic methods to generate new data from existing Australian cohorts with deep clinical or functional data, using approaches like the characterization of human knockouts to discover and validate new therapeutic targets, and then finally continuing the work that my group had done in Boston to apply a variety of genomic and informatic approaches to improve the diagnosis of rare diseases in collaboration with a large number of existing groups, particularly in Melbourne at the MCRI site. And just a final slide, just to mention something about our flagship project, which is the construction of this genome reference database. The driver for this is that we need to come up with a way of better serving the rare disease patients in Australia and elsewhere who come from communities that aren't currently represented in reference databases like gnomAD. And that's a real problem because it means when we interpret the variants that we find in those patients, we can't actually determine whether those variants are true potential disease-causing variants or common variants that just aren't reflected in the European-centric gnomAD dataset. So to fill that gap, we need to do a better job of fleshing out the reference databases with Australian diversity. So our goal here will be to recruit 10,000 or more Australians from diverse communities, focusing on the groups that are neglected in existing databases, and that will ultimately include Indigenous representation as well. And that will be done through ongoing relationships with communities to build up trust and the opportunity to recontact those groups for future research. For those individuals, what I would like to be able to do is to do whole genome sequencing on everyone, as well as generate transcriptomic or potentially other functional genomic data from single cells from blood, and then ultimately make that data as quickly and as widely available as possible to the research and clinical communities, particularly to the clinical genomics community, to fill the need of improving the diagnosis of patients from those communities. So if you're interested in participating in the centre, feel free to contact me. My email address is available online, and there's a website up for the centre as well. So with that, I would like to finish by thanking everyone who's been involved in putting the gnomAD dataset together. There's a big list of names here who've been involved in making this possible, including our analysis team and mini groups, particularly the Broad, who've been involved in coordinating that effort. My lab back in Boston, who was involved in many ways in our rare disease and gnomAD work. And then big thanks also to the 109 principal investigators who've allowed their data to be used in the EXAC and gnomAD datasets. Thank you guys very much for listening.
Thank you very much, Daniel. I'll clap on behalf of everyone. That's something that doesn't quite translate to the Zoom.
Yeah, it's difficult. That's right. Yeah.
So we've had a lot of questions come through. So if you're happy, we're just after the hour. So if anyone needs to leave, of course, please do so. But we'll stick around and work through these questions for anyone who can and would like to stay. So first one I'll go back to from Gokchen Razlan again, who's asking about what might be the function of non-housekeeping genes on the red side of the spectrum. So on the, I guess on your loss of function spectrum, do you have any comments on what else is likely to be what's falling in that red spectrum?
We've taken a look at them. I mean, a number of them are the obvious genes involved in core cellular functions, things like transcription, translation. There's a whole host of genes, though, that are not cellular housekeeping genes, but are involved in critical developmental functions. Some of those are well established; they're well known to the developmental biology community. Some of them are clearly transcription factors and other things where the role is not yet well understood. And I think there's a lot of work still to do for at least 3/4 of the genes in that data set. We actually don't have a well-established function. And so it's clear that would be a group of genes where really focusing our efforts both on disease gene discovery and also on functional characterization will be really worthwhile. I think that in general are going to be a very important set of genes.
Cool, thanks. So Emily says some loss function mutations don't result in complete nonsense-mediated decay, whereas some do, and some have additional impacts on splicing. Were these sorts of factors taken into account with respect to the loss of function analyses you've presented?
Yeah, to some extent. So we do, as part of the filters that we apply through Lofty, we do take into account whether or not the variant is likely to trigger nonsense-mediated decay. So variants found right at the end of the gene where it's both first unlikely they trigger NMD, and secondly, even if they, and the resulting impact is likely to be a very, very small truncation of the protein, those are excluded from our high confidence set of loss of function variants. And in the splicing case, there's some real subtlety there because you can, for instance, have a predicted splice disrupting mutation where there's a nearby site that can just sort of take over if that occurs and keep the transcript in frame. Lofty does have some tools in there that allows us to predict that, but most of those cases we end up having to catch through manual curation. And that's one of the things that our curators look at when they go through these variants is the possibility of a rescue splice site nearby.
Cool. Dr. Nandita Puri asks, regarding the results from the loss of function analyses, can you comment on the utility of bioinformatic prediction of quote-unquote pathogenicity of missense variants like SIFT and so on, mostly based on conservation, and how well do they correlate to what you've observed?
So we can use gnomAD to assess how well those factors perform. So there's a measure that we built called MAPS, which is basically a measure of the proportion of variants in a particular class that are super rare, that are singletons in the gnomAD dataset. We have to adjust that for mutability. But that actually gives us a pretty unbiased estimate of how deleterious each of those classes of variation are. And it's clear that there is some information contained in those missense pathogenicity predictors, but they're not that. The missense variants that are predicted to be highly pathogenic, even using those scores, still don't come close to being as deleterious as a true loss of function variant. So I think they're helpful, but you have to interpret them obviously with a real grain of salt. They carry some information, but not too much.
Yep. Paul Lacars asks, how do we scale manual variant curation to more than 500,000 samples? Is this feasible? Is it essential? Or can we do studies, or what studies can we do without it?
That's a great question, Paul. And one we've definitely thought about and keeps me awake at night, frankly, because it's clear that you do actually, before you really sink serious effort into following up a particular variant, you need to do manual curation. The automated filters we have just don't catch many of the classes of error that exist out there. So a couple of things that we've thought about here. The first is we built a portal for doing that curation, so that and that basically just pulls together a range of different types of information, data from the UCSC genome browser, data from gnomAD, and a few other sources into one place, so that a curator can very quickly just go through that type of information and curate with an eye towards whether or not something is a loss of function variant. And then associated with that, we have an evidence ontology, so you can then click, the curator can then click very quickly whether they think it's real or not, and also mark off the types of evidence that they've used to exclude or include that variant. So I think that'll help. That allowed us to do about 4,000 variants over the course of a month or so with one curator. But that's scaling that up still requires significant amounts of work. I think we just need to think harder about, firstly, doing a lot more manual curation and experimental annotation of loss of function variants, using that to train better predictors. And at the moment, many of our approaches are still pretty heuristic. Machine learning is going to be a major contributor here. It's just that we don't yet have fantastic training data to really get that together. But machine learning will take us a long way. And then once that first step filtering is improved, we can probably get rid of a large tranche of easy errors. And then after that, we'll still need to have a set of variants where we go through manual curation, but with decision support. That's the current plan. Whether it will scale to 500,000 exomes remains to be seen.
Thanks. Liam Fernley asks, can you comment on how cross-platform differences are handled within the gnomAD exact data aggregation steps? Problems around things like duplication marking have impacted projects like UK Biobank's large-scale whole exome sequencing efforts. How much of a challenge was dealing with these in standardizing all the data?
Liam, actually, I don't know if you could say directly, do you mean, for duplication marking, do you mean reduplication or duplicate samples?
So if you can unmute yourself, Liam. And
I think that's working.
Yep.
So I'm referring to how they've had to basically put a hold on all their WES data through one of their pipelines with the reduplication around pipeline issues in the NovaSeq flow cells. And then there are now questions about the functionally equivalent pipelines that they've got in there. Really, you've got a lot of data coming from a lot of platforms, and I'm just wondering how that challenge of aggregation is handled.
Yeah, no, totally, that's right. Thanks for clarifying that. That's really helpful. And clearly Regeneron has really struggled with the analytics of that data set. That's caused some real delays in the UK Biobank exome release. But it's a generic set of problems. The one thing we've been able to do is, at least because all of the underlying data that flows into gnomAD gets reprocessed from the read level. So we put it through the same processing pipeline. It all gets realigned, processed the same way. There we were very fortunate because we could take advantage of the fact that most of the data had already been sequenced at Broad. That meant that most of it had already gone through that type of approach. So that means that we can still have errors in the underlying processing, but at least those errors are uniform across all the samples and we don't have batch effects. So that's very helpful. I think that there's little other ways to solve that other than ensuring that you have your underlying samples go through the same pipeline, or at least as closely aligned a pipeline as possible. And that experience and many other experiences have shown us that even relatively subtle differences between the pipelines can be enough to throw things off. Functional equivalence really does help here, though. At least there's a set of standards that we can use to define getting right up to the step of having a cram file that's reasonably well aligned, and that has helped us a lot. And we have been able to, in a subset of projects, not in gnomAD though, we have been able to bring in some external data that's functionally equivalent crams, combine those together with our data sets, and that's usually worked reasonably well. So that certainly does help.
Great. Question from someone whose name is a random string of letters. What are your plans for Indigenous genomics? And I guess a quick one also attached to that is data sharing from Population Genomics Centre will be via the cloud?
Yeah, at this stage, I don't think I can give you a well fleshed out description of the plan for Indigenous genomics, except just to say my sense here is that this will have to be a long conversation, and we're not in a rush to move that part of the project forward. We want to start the discussions now, but the anticipation is that it's going to take a long set of discussions, both with Indigenous communities and also with other groups that already have a lot of experience in that space, before we're in a position that we can do any sort of large-scale genomics in those areas. But at the same time, it is critical that for any sort of Australia-facing reference database that we do have representation from those groups. So we'll start the conversation now, and then we'll see where that goes. For data sharing, yes, the plan would be for that to be through the cloud. The details of that will depend on exactly which platform we end up going for. But the idea would be that we have a single shared repository that makes it as easy as possible for other people to bring their compute to the data.
Great. We'll try and wrap this up. So by 1:15 and start the let the ECRs have a full turn. So there are 3 remaining questions I'll put to you quickly, Daniel. Richard Edwards asks, how much of gnomAD's successful update do you think is down to the unique data set, and how much is due to the time and effort that went into the interface?
Good question. I'm not sure we really know. So early on, in some ways, this success was easier because it just was the biggest data set that anyone had at the time of exomes and genomes together. So we put a lot of effort into the interface just to try to make it as easy to use as possible. But I think it still would have been fairly heavily used even if that hadn't been the case, just because there wasn't another comparable database out there. I suppose we have a few data points since then. So there have been other large-scale resources that have been put together. So Bravo, for instance, which is a browser that provides access to the TopMed data set, they certainly do have a significant number of whole human genomes. There, though, there's been real issues with people getting access to the underlying TopMed data set. The Bravo browser has things like it requires a Google login before you can actually use the browser. And there's some other restrictions to the way it gets used. And I think that has resulted in dramatically lower usage of that browser compared to the gnomAD browser. So clearly UI and accessibility makes a big difference in that space as well. But I think it's hard to untangle. I don't know what the real answer is.
Thanks. And Harriet Dashno asked a very appropriate question, given her
Yeah.
work in the past. What are you planning to use for SDR variant calling on the upcoming gnomAD release? Can you comment on challenges, for example, accuracy?
Not with any great accuracy compared to the sorts of comments that Harriet could make. So I mean, the plan there is Ben Weisberg's been within the team at the Broad has been looking at a variety of different SDR tools and trying to compare some of their accuracy. I'm actually not fully up to date with how that benchmarking is going, but the plan would be based on a combination of the relative accuracy, sensitivity and specificity of those tools, but also the scalability, how easy it would actually be to run those on 70,000 human genomes. We'll then be able to make a decision about which set of tools are feasible to run. Harriet, I don't know if that group's already been in touch with you, but if not, I'd be more than happy to facilitate discussions because it would be great to get your input before any final decision was made.
That'd be great, thanks.
Cool. Okay, finally, second one from Richard Edwards. How many whole genome sequences do you think we need before we can give every amino acid a constraint score based on varying frequencies akin to conservation scores? I've seen this done at the whole protein/domain level, but when can we zoom to specific residues? I think you were close for CPG codons. Yeah,
great. And Richard, you're ahead of me. For CPG dinucleotides, as you've noted, because the mutation rates are much higher at those sites, we actually are getting close. Even in gnomAD, we were starting to get to the point where we're seeing some saturation of discovery of those variants. So we're not far away from being able to say for any given individual methylated CPG site in the genome, with the next release of gnomAD, I think we'd be at the point where we could say the fact that variant isn't present in the data set is indicative that that's probably lethal or deleterious and therefore removed from the population. But for other sites, CPGs are the best case and they have an incredibly high mutation rate. For most other sites in the genome, it's going to take us a lot longer to get there. I can't remember Konrad Kajewski could certainly give a much better informed answer to this than I have. but my sense was certainly that we need to be in the tens of millions of samples before we can start to get to that. And then there will be a subset of very slowly mutating residues where it's going to take even more than that before we can start pulling that out. But it will be interesting, I think, for things like being able to say, for particular stop codon residues that are CpGs, we're going to get well ahead of that. And so there'll be particular questions we'll be able to answer at different stages in that sample size growth curve.
Great. Thanks a lot, Daniel. Thanks everyone for the excellent questions. Thanks, Daniel, for a great talk. So much exciting work described there, and lots and lots to look forward to as well, with the new center, the new teams, and building on everything there. So with that, we'll